We in Manchester have done a lung health check pilot where we've taken screening into deprived communities where there's a high prevalence of lung cancer, and we do it on like a truck in a supermarket car park, and it's worked really, really well. So you get lots and lots of people coming, and a lot of them are high risk, and they undertake their scan, and we found quite a high prevalence of lung cancer. Trying to then integrate sort of biomarkers into that high throughput process is going to be quite tricky. But we have achieved it. So in our second round, we did something called the Community Lung Health Study, where we asked people who came to have a blood sample. And these people are from very deprived areas. You know, the sort of form book says that they won't take part in research, but they absolutely were very helpful, really accommodating, and more than 80% of people signed up to take part in a biomarker study. So we think that we have a model, we can access people who are high risk, who are very willing to take part in biomarker research. So we think we have a platform. The next thing then is obviously to develop a biomarker that actually works. So there are challenges, there are different questions in early detection of lung cancer. One is, can a biomarker actually detect the cancer itself? That's going to be really challenging because we want to detect cancers when they're only tiny. So trying to detect a molecule from something tiny in a body is exceedingly difficult. So there's another way of doing it, which is actually to do a biomarker where we look at lung cancer risk. Because the way we select for screening is by assessing someone's risk. So when they came to our van in our supermarket car park, we asked them a series of questions, and those questions led to a number, a lung cancer risk number, and if you're over a certain threshold, then you were eligible for screening. And really the way I see a biomarker would just be another variable added in to that risk assessment. So whether that's looking at your genotype, are you susceptible to the cigarette smoke, whether it's a marker of damage to the lung, whatever it is, if it can go into that risk stratification model and make it more precise, then we know from the research out there already that will increase cost effectiveness, reduce the number of screens we have to do, and actually detect more cancer. So it makes the whole process better. The secret is actually finding the biomarker that actually will do that, and that's what we are intending to do. It took a lot of collaboration, a lot of close working. What you have to do is just it's a multidisciplinary team. It's not one person sat in an office somewhere. So you have to bring in your clinical team, your research team, bring everybody together, and just figure out the logistics of it. So you have to, obviously, the first thing is having the patient population coming through, so that was the clinical service, and then building on top of that, well, what do we need? We need decent samples, really high quality samples getting into the lab. It's no good us collecting samples and them being poor quality, because then the lab can't actually process, can't analyse them, it's poor quality. So right at the very beginning, everybody sits around the table, what do we need to do? Every part of the pathway needs to be as perfect as possible, and then you're really giving the scientists the best opportunity to find that biomarker that will actually make the difference.